Bakshi from University of Kollani, who very well explained yesterday the usefulness of such exercises, specifically when we are dealing with in silico data of the living system. Good morning, Dr. Bakshi, and welcome to the second day of the workshop. As you know that Dr. Bakshi is a biochemist turned into bioinformatician and a computational biologist, and has already given the hands-on workout with the various biological databases yesterday. Today, he will be covering the aspect involving sequence analysis, which is the process of subjecting DNA, RNA, or protein sequences using some analytical methods, and further to understand their features, function, structure, or evolution. But before we start his session today, I am extremely happy to announce that we have our respected Honorable Vice Chancellor, Sir, Professor Dr. Soikot Moitro among us to address the audience. Sir, we are extremely honored to have your gracious presence today. I will now request you, sir, to address the participants and enlighten them with your thoughts and projects for such endeavor. Welcome, sir.
Okay. Thank you, Dr. Pati. Yesterday, I was supposed to join, but because of this network failure, I could not join. And whenever I got the chance, I thought that it would be too late to join the proceedings because those lectures and all these things had already started. Did you invite anyone from Kashmir IUST to be a part of this panel discussion?
Yes, sir. I also requested BC sir of IUST.
Okay, okay.
But he is now busy.
Okay, okay.
Sir, actually, I just want to share that Professor Siddiqui, he actually wanted to join, and he said, if I get time, I will just enter for 5 minutes, because he has a selection committee from yesterday till today. But he said he would. So
Professor Siddiqui's presence would be a great thing, because
yes,
he is a man of this background, biotechnology, biology,
yes, sir.
biological science, and he can throw some interesting, this aspects for relevant for this workshop. Now it's a great thing that from Bioinformatics department, Dr. Poti has taken this initiative in organizing this workshop. And I think that maybe next time you can extend this time duration to make it a full-fledged faculty development program. To talk about bioinformatics, so far though I do not belong to this discipline, but from my general understanding, I'd like to say a few words. It's basically an interdisciplinary field of science which combines biology, computer science, information technology and engineering, mathematics, and statistics for analysis and for interpretation of biological data. This has been used for in silico analysis of biological queries using mathematical statistical techniques. Now, this emergence of bioinformatics, it resulted in understanding biomolecules at the molecular level using various visualization softwares, machine learning, understanding evolutionary genetics, DNA sequencing, drug development, precision medicine, data analysis and interpretation. So it's from in the last 2-3 decades, it has become an upcoming field due to information technology, and particularly in this decade because of this revolution of artificial intelligence and machine learning. This this growth and development in artificial intelligence and machine learning, handling of big data, Hadoop platforms, all these things, making this field more and more, this exciting. And if you look at the market, So this is the report from Industry Daily Observer, the global bioinformatics market. It is in 2017, it was more than $6,000 million. And by 2025, it is going to be more than $18,000 million, almost 3 times growth will be there. And if we look at the compound annual growth rate from 2018 to 2025, it is almost 25%, 13.88%. So this world bioinformatics market, now it is segmented based on technology, service, applications. Now with this development and emergence of computational biology, health science, genetics, genomics, drug discovery, and molecular biology, it has created a huge scope for life science industry professionals. And this is going to be one of the biggest future for aspiring researchers in the field of life science. Now, if we want to build up good career in bioinformatics, as it is interdisciplinary fields, it requires both expertise in computer science as well as biology. We need to have programming skill like C, C++, Perl, Shell, Python, with knowledge of Linux systems. Then R program, statistics with R programming. Then bioinformatics database like NCBI, Entrez, Uniprot, Swissprot, MySQL, PDB. Toolkits like Scaffold, Protmax, Indigo, SwissDoc, OECAM, and others. Data mining techniques, how to extract data, data analysis, data visualization techniques like visualization of the extracted data using different simulation, understanding of molecular biology and genetics. And for gaining knowledge in all these areas, it is not necessarily you have to invest a lot of money, because there are large number of MOOCs courses available. I personally have seen the courses, although I have seen it from the surface only, but you can always go into the depth and can gain meaningful, this knowledge and ideas from websites like edX, Coursera, Udacity, and there are large numbers of open courseware. So gaining right skill and knowledge is now not a difficult proposition. So with this, only you have to develop the aptitude and liking for these subjects. And I believe that it is going to be the subject of the future. And in India, considering the fact of this huge biodiversity and huge volume of data, biological data available here, there is a tremendous potential of bioinformatics in coming years. And already different organizations, they are showing interest for investment in this research in bioinformatics, computational biology areas. Many IT organizations, many pharmaceutical organizations, they are investing lots of monies. So it's now a growing field. And my expectation that from our university systems and from our, these training systems, will be able to generate a large number of professionals to cater the need of future, the requirement in this particular field. So with this, we have this department. We want to make it this more active, more effective in imparting knowledge and carrying out researches, training in bioinformatics for the benefits of large number of learners. And I sincerely believe that from yesterday and from today's hands-on of this this deliberation by our resource persons, the learners will be benefited. But of course, this today's workshop, it's just something like a curtain raiser, or maybe tip of iceberg. Now you have to take all the participants, you have to take active interest in this field, and self-learning is the best way of learning. And as I said earlier, there is no dearth of resources in this digital world to enrich yourselves with the right set of knowledge and skill. With this, I wish this workshop a grand success, And I wish a great career and successful life for all the participants who are present over here. Thank you all.
Thank you very much, sir, for your ever encouraging and motivational speeches. The participants will surely get encouraging experiences from your advice and more particularly from the training they receive from this workshop. Thank you once again, sir, for your kind presence today. Now I will request our director, Professor Raja Banerjee, to say a few words on today's session and encourage the students and faculties for taking part in this workshop. Welcome, sir.
Good morning to you all, especially to our Honorable Vice Chancellor for his kind presence today and the motivation speech. Actually, yesterday I told you, I have showed you also one slideshow through which I wanted to tell you why we should learn the bioinformatics. And basically, based on that, already Dr. Angshuman Bakchi, I introduced him yesterday also. So he will tell you, he has given you a glimpse of the what is the basically the bricks of the thing. That means now you have the amino acids sequence, the DNA sequences, and how to get this idea, how to take this, and now you have to, from the pond you have to go to sea. So I think today I shouldn't take more time. I think Dr. Bakchi is already ready with his things, so he will now today take you to the, if not the deepest part, at least to the surface of the ocean, where you can see how the bioinformatics tools, this sequence database and etc. are utilized to get more prediction knowledge. So yesterday he has told you, I have also told you, so it is not possible for all of us to do all kinds of experiments. Working, isolating one protein, isolating one DNA extract is very difficult. Analysis part is difficult. But if you know the sequences, so it is called the basically structure. Initially it was called structure function relationship. Nowadays in one of my paper I use this term sequence structure function relationship. So if you know the sequence, it is like just a bottom up approach. So if you know the sequence, from the sequence, if you know some homologous things, you can get the template, you can have an idea of the structure, and you, once you have the structure, you can predict the function. So the function is a very important thing. If you know the Alzheimer's disease, this is the misfolding. If you consider the Huntington's or other diseases, that is also misfolding. So for proper folding, you need the proper structure. If for knowing the proper structure, you know the proper sequences. So with this, I just request Dr. Shobhan Poti to start the session today. Thank you all.
Thank you very much, sir, Professor Banerjee, for your valuable words and views on the workshop. We also expect that the students will be benefited from this workshop. Although it would have been better to conduct more such sessions, we will surely take initiative to hold these kinds of training in the near future for the benefit of the students and researchers working in the areas of computational biology and bioinformatics. Now it is the time for today's session to start, and I request Dr. Angshuman Bakchi to take over the session and start his lectures for today. I will also request the audience to keep your computer or laptop ready for today's exercise. So Dr. Bakchi, you may now start your session formally, and all the very best from the entire team of the MAKAUT for a great session ahead. Thank you.
Request Dr. Joya Bandyopadhyay to say something. He is there.
Yes.
Good morning, sir. It is a great honor for me also. As you have seen that I have registered specially to take lessons, additional lessons from whatever we have learned all through. And Dr. Bakchi has done an excellent job since yesterday. And it is also a great, it is my honor in a way, because this is the first time I'm attending any computational biology workshop. Thank you, sir, because it is only because your encouragement and your drive to make these sort of seminars now more on the online platform has made it more convenient for all of us, all researchers, faculties, to have a very a kind of a global idea about all the recent researches going on, and especially from the basics to the high-end research. And Dr. Bakshi, although I have been introduced through this workshop for the first time, and it is very good that he is just coming from our neighborhood from Kalyani University, and we will definitely have a great time ahead in the days to come with more and more of researches in the areas of bioinformatics. And definitely, as Professor Banerjee has pointed out, that it is a living system, and with the DNA, RNA, protein, and sir, you have also pointed out that without all this, the entire scientific field doesn't only focus onto one particular field. It has all varied types of scientific subjects that we can ponder over and make our new generation have this flavor of many such interdisciplinary subjects, and bioinformatics is one of them. And though I belong to biotechnology, we require this subject every day of our research. So I cannot think this particular training not helpful for me, because it is very much indeed a part of all the biological science researchers. So I do not want to waste any more time. Thank you very much. You have, sir, that you have asked me to speak. I feel very honored, and of course I'm registered, but I'm not a panelist. But nevertheless, sir, it feels good to talk on behalf of the department for the university and also for the faculties. So thank you very much, sir. And it is very good to see you today. Yesterday we missed you, and I think Shomin has done an excellent job in moderating the entire session, and we are having a very good session. So trust me, sir, we will definitely deliver in the days to come with more such sessions. Thank you so much.
Thank you so much, Dr. Joy Bandyopadhyay, the head of the biotechnology of our School of Biological Sciences. Thank you for your motivational and valuable speech. Thank you so much. Now I welcome Dr. Bakshi to start today's session.
Am I audible?
Yes, sir.
Okay. So very good morning to all of you, Honorable Vice Chancellor, sir, Professor Shoipat Mitra, Professor Raja Banerjee, the Senior Professor, Head of the Department, Professor Jaya Bandyopadhyay, and the Coordinator, Dr. Shomin Poti. So very good morning to all of you. So today, this is the second day of this workshop, and today I will be talking about the analysis portion. So yesterday, we learned a little bit about the different databases that are mainly used to solve the biological problems. So as Professor Jaya Bandyopadhyay rightly said that this is bioinformatics is related to biotechnology, since biotechnology deals with the obtainment. That means biotechnology deals with the analysis of DNAs, RNAs, proteins, and all those biomolecules. Bioinformatics, the main aim of bioinformatics is to aid in the process of the analysis. I mentioned earlier, yesterday as well, that these techniques, that means the wet lab biology techniques, these techniques are very much expensive and as well as labor intensive. So what we, the bioinformaticians, do is that we try to streamline the entire process so that by performing not a multitude of experiments, but some experiments, we can come to a conclusion about a biological problem. So that is the basic aim of bioinformaticians. We predict the pathway. We help the wet lab biologist to streamline the entire experimental design. So with this background, let me continue where we left yesterday. So I have, let us have an interaction with the participants. Participants, if you have any queries from what we learned yesterday, you can clear it up right now so that we can proceed to the next level, that is the analysis portion. Because yesterday we talked about the data, the databases, the data that are present in the databases. Today, we will be using those data and we will try to extract as much information from the data as possible. So participants, I request you to clear up your questions, your queries from yesterday. If we don't have any questions, then I'll move on to today's topic. So participants, you can write your questions in the question answer box or in the chat line.
You write your question in question and answer box only, not in the chat box. Only
Yes. Any question in the I don't find any open question, but
sir, you may start today's session.
so let me share my screen with you. So is the screen visible to all of you?
Yes, sir.
Okay. So today, what I'm going to talk about is what we have the databases. We have the data present in the databases. Then what we can do with the existing data. See, yesterday, when I was talking about the NCBI, I showed you that NCBI contains nucleotide sequence as well as the amino acid sequences of proteins. Now, and basically, by taking the sequences, what we try to do is we try to extract as much new information from the sequences as possible. So there lies the importance of sequence comparison. From protein sequences, what we can do is we can identify the similar sequences. The similar sequences may be called the homologous sequences. And those sequences are supposed to have similar kinds of structure and function. We can then analyze the three-dimensional structure of the protein that is made up of the amino acid sequences present in the database. We can construct phylogenetic trees to determine the evolutionary history of the sequence. We can perform the various annotations of the entire genome. And finally, from these results, we can identify the specific regions where we can fit in any suitable drug. That means for drug design purposes. These are the basic importance of sequence comparison methodologies. Now, by sequence comparison methodologies, what I want to mean is the analysis of amino acid sequences of proteins. Now, the question should arise here: why proteins only? Why not nucleotides or the nucleic acids? We can perform a sequence comparison of the nucleic acids as well. But as I told you earlier, and also Professor Raja Banerjee mentioned in his short presentation yesterday, that proteins are the central players of the biological processes. That is why we mainly focus on the analysis of the protein sequences. The protein sequence analysis can be performed mainly in two different ways. The first one is pairwise alignment. As the name implies, it is the alignment of two sequences. And the second one is the multiple sequence alignment, alignment of more than two sequences. For this entire process, today we will be using the tool BLAST. Now, here I would like to mention that these tools are dependent on a complicated algorithm. The basic algorithm is for sequence alignment, that means pairwise alignment, is the scoring of the sequences. We will learn in the later portion. And for multiple sequence alignment, it is called the dynamic programming method. So, first is pairwise sequence alignment. Pairwise sequence alignment, what it does is that it tries to compare an unknown sequence with the known sequences that are present in the databases. This is in a simple way. For example, in the last class yesterday, I showed you that in the databases, that is NCBI or SwissProt, you deposit the data of nucleic acids and proteins. Now, say from your lab, you identified the amino acid sequence of a protein. I told you yesterday that in order to get your paper published, you have to deposit it to the databases. So what you do is you get the sequence and deposit the data in the database. Now, the next thing is a mere sequence does not provide any important information until and unless you have the annotations. I showed you the different annotations yesterday. Those annotations come from the comparison of the sequence. First, we perform the pairwise comparison. What we do is we take the sequence, the unknown sequence that we deposited in the databases, and then we perform a pairwise alignment. Pairwise alignment means we perform one-to-one alignment of the unknown sequence with the known sequences that are present in the database. And we get several such alignments. On the basis of that, on the basis of those alignments, the next step is to choose the homologous sequence from the alignment. Now, say for one unknown sequence, you have 100 pairwise alignments of the unknown sequence with the known sequences that are present in the database. So, which alignment would you choose? The basic reason is the alignment to choose should have a very high score, that is called the identity score. And those identity scores are coming from the matrices, the PAM and BLOSUM. These are the matrices which store the scores for the pairwise alignments between the amino acid residues. There are certain other aspects as well, like gap and the significance score. The gap is a region of mutation. That means while performing a sequence alignment, we may have a sequence alignment where the two sequences are not aligned. That means you might encounter a situation where in the alignment, there are certain regions where no sequences are present. Those regions are called gaps. I will show you, and this portion will be clear when you will do the hands-on portion. And the significance score. The significance score gives you an idea about how reliable the alignment is. For example, let us consider a very crude thing. Say, in your lab, you identified the amino acid sequence of a protein from a prokaryotic organism. While performing the sequence alignment, it might happen that you get a very good alignment with a protein from human. It might happen. But this is not likely. This is unlikely. A prokaryotic protein should generally have similarity with another prokaryotic protein. I'm not saying that it is absolutely impossible to have that alignment, but that is unlikely. So, the reliability of the significance score gives you an idea about how that type of alignments are handled. When there is some kind of mismatching between the natures of the organisms. Now, the next thing is the scoring. This is very important, I told you, because on the basis of scoring only, you will be able to identify the homologous sequence. So, for scoring, what we consider is first is identical sequence pairs. For example, both the unknown and the known sequences have the same amino acid residues at the identical position. This is called identity. Then comes synonymous substitutions. For example, in the unknown sequence, there might be a V, which is substituted by an isoleucine or a leucine in the known sequence. Both V, that means valine, and isoleucine or leucine, these are hydrophobic amino acid residues. So this chemical similarity between the amino acids to point towards the fact that though this is a substitution, though this is not an identity, but still this is tolerable. You can tolerate that. A hydrophobic amino acid may be substituted by another hydrophobic amino acid. This is called synonymous substitution. Now, when a V is substituted by an aspartic acid D or a glutamic acid E, in that case, it is called non-synonymous substitutions. Such cases are generally not considered to be a very good alignment. And while scoring such alignments, the identical or the identities are given the maximum score. The synonymous substitutions are given a little bit lower score, and a non-synonymous substitutions are given the lowest score. The similar scheme is followed for nucleic acids as well. So here I'm showing you an example. See, this is ILLI. This is a synonymous substitution. This is an identity C. This is an entirely identical. AS, alanine, serine. Alanine is hydrophobic, serine is polar. This is a non-synonymous substitution. These regions are called gaps. Yes, any question? Any question? I heard a sound, so if you have any question here, you can ask me.
Sir, no, sir, please proceed.
Okay. So I was talking about the sequence alignment. So here I'm showing you, this is the exact identity. This L substituted by I or I substituted by L, this is synonymous. This is non-synonymous. And these are the gap regions. These are the gap regions. So all these substitutions are given some specific scores. And as I told you that the scores are coming from these matrices, Blossom and Pan. So this is an example of a substitution matrix. See, this is a set of amino acid along this row, along this column, the same sets of amino acids are there. So what the matrix shows is that A matching with A is getting a score of 4. A matching with R is getting a score of -1. A matching with L is getting a score of -2. A matching with D is getting a score of -2. A with C, 0, and so on and so forth. Similarly, R matching with A, getting a score of -1, R matching with R getting a score of 5, and so on. This is basically the substitution matrix. And from this substitution matrix, you can have the scores for the alignment.
Dr. Bakchi?
Excuse me?
Yeah, can I ask you one simple question over here? Since we are dealing with the matrix, I would like to ask you one question.
Yes, please.
Yes, yes. Just since this table is there, and I can see that, I may have missed it out in your previous slide, but still I would like to
Madam, your
clarification
voice
here.
is breaking. Could you please be a little bit loud?
Yeah, I am loud. Am I audible to all?
Um,
Can you
Madam,
hear
it's
me?
better that if you could write the question here,
Okay.
because I think the
Okay,
voice
okay.
is breaking.
Fine. Then I will write the question. You can proceed. You may proceed. I'll write the question.
Okay, but Madam,
Hello?
we can discuss. So,
Yeah. No problem. You please go ahead. I'm writing the question. Okay.
okay,
Fine. You
okay.
go ahead. Yeah, sure.
See here how to deal with gaps. Here, no gaps are present. So you are getting a score like this. And this PID means the percentage of identities. So you see, this is one identity, this is another identity, this is another identity, this is another identity. So 5 identities out of 7. Now, this is a kind of a dot matrix method. That means how to align the sequences. But this is this kind of gives you, in this particular case, what you do is you write the sequences and then put a dot where you get an identity. And that way you do a trace back. Like if you move from here, you can do the alignment like this. But these things are already present in the tools. So the bioinformaticians, since we are more interested in results, so let us perform an alignment. Then I will again come to this. So let us do a hands-on thing so that the concept would be a little bit more clearer. Participants,
Okay, sir.
let us do a small alignment first. So participants, I would tell you to first go to NCBI. Now, go to protein and type the accession number that I showed you yesterday. You complete up till this step and then we'll proceed.
Okay, sir.
It's done.
Yes, sir.
Okay. Then go to search. We got this.
Yes.
Let us take this one. Click on the protein ID. Let us get the faster sequence. This is the faster sequence. Then again, open NCBI. Go to BLAST. This is called Basic Local Alignment Search Tool. We perform up till this.
Yes, sir. Sir, we just need to wait a few seconds for every participant can get this up to this. because network connections are not that much strong everywhere.
Sorry, I could not
Sir,
hear you properly.
sorry, sir. I just request you to just wait for a few seconds for every participant can get
Okay,
up to this.
I'm waiting. I'm waiting. No problem. No problem.
Okay. Okay, sir. Thank you, sir.
Just let me know when everybody is done.
Okay, sir.
Dr. Bakchi,
Yes.
can you just check the chat box? I just kept the question in the chat box.
Okay. Okay. The scoring is 4 or 5. Okay. Yeah, I got your question, madam. The scoring is 4 or 5, even when the same when paired with the same amino acid. This is a very important question, madam.
Yes, please, sir.
These scorings are coming from aligning similar amino acids, that means similar kinds of proteins. For example, say you have taken a set of proteins which are all DNA binding proteins having helix turn helix DNA that motif, helix turn helix DNA binding motif. Now what you are doing is you are aligning that particular motif. During that alignment, the number of times you are getting alanine at the identical position, you are taking that as the score. So it might happen that out of 100 alignments, you are getting alanine at the number 1 position of the helix and helix motif for the 95% of the time. The score is calculated on the basis of that frequency.
Okay, okay, I got it. Thank you.
You're welcome.
Okay, sir. You may proceed,
Okay.
sir. Everyone got up to this.
Everybody is done?
Yes, sir. Yes, sir.
Okay. Now, since we are using protein, so click on this, protein blast. Okay, all of you got this?
Yes, sir.
This window?
Yes, sir.
Now, copy this FASTA sequence and paste it here.
Okay, sir. Done, sir.
It's done.
Yes, sir.
Done for everybody?
Yes, sir.
Now, you can choose the database. Let us consider the PDB, protein data bank. Let us choose.
Okay, sir.
And this is BLAST P. P stands for protein blast.
Okay, sir.
Okay?
Yes, sir.
Now, let us play with this algorithm parameters. I will explain everything, but here you are exposed to this matrix. So here by default, the Blossom matrix is present. You can change it, PAM, anything, but let us keep it to Blossom.
Sir, I have a question. May I ask you?
Yes.
Sir, what is the specialities of this kind of matrix?
What
Like
is the importance of?
Importance not, sir. What is the speciality of this categorical matrix? Like bosom and
Okay. The two matrices were built in different ways. The PAM matrices were built by performing or by comparing sequences which are very similar.
Okay,
and Blossom matrices were obtained by performing alignments between sequences which are highly dissimilar.
Okay, sir. Thank you, sir.
So different scores are present in the alignment in the matrices.
Okay, sir.
So
But sir, here I have one more question.
yes,
Before
yes,
doing
go ahead.
before doing any kind of blast, how may I know the sequence alignments are similar or dissimilar?
Right. That is why it is always better to use Blossom, because even if the sequences are very similar, in that case, Blossom would consider it to be dissimilar, and the dissimilar sequences, it is really difficult to analyze dissimilar sequences. So first, everyone should start with Blossom, because we generally do not know that whether the sequences should be similar or not. So that is why Blossom is used. If we have idea that, okay, my sequence belongs to this particular family, then only we can use PAM.
Okay. Thank you, sir. Thank you so much.
Dr. Bakshi,
Yes.
this is Raja Banerjee. One thing, as you are showing, there is a Blossom 62, there is a Blossom 10, etc. I will be thankful if you just
Yes.
mention what is the value of this 10,
I will
62,
do that. I will definitely
and
mention that, all those stuff.
yeah,
But let me explain
yeah.
and this sequence alignment portion. I will
And
definitely come to that.
also,
I will
also,
definitely
also,
come
also,
to those alignment algorithms parameters as well.
and also the PAM 60. Suppose Blossom 60 and Blossom 100, and PAM 60 and PAM 100. Which one is
Yes,
greater?
I will explain everything.
Okay, okay, okay.
Okay. Now everybody got it? Got the results?
Yes, sir.
Everybody?
Yes, sir.
Okay. So see, this is kind of your result. This is the score that I was talking about. This is called the query coverage. That means, see, total amino acid length is 121. Among the total 121, only 79% got aligned with an identity of 50, nearly 57%. And since we used the PDB database, this is the PDB accession number. And this e-value, this is the expectation value. You need to remember that the lower the e-value, the better is the result.
Okay, sir.
So this way, the alignments are given. Now, let us see the alignments. This is called the graphical summary.
Dr. Anshuman, can you go to previous slide?
Yeah.
Yeah, actually, if you see that there are two values, okay, 79 suppose you have a sequence and the maximum score is 115, both the sequences are there, 115, 79%, and these two are identical, because this is 608 LK, etc. So that means this is a similar protein, isn't it? If the, as the
These two are
as
kind of the similar. That means all these two.
yeah,
These
yeah, yeah.
two, I believe that these two are the same proteins having different PDB IDs.
Yeah, and if you come to just this is one query. If you come to the next one, that is also showing the percentage is 57.29. Okay, but if you see that I value, sorry, e value is less compared, is higher compared to that of these two. So if anyone asks you which one will be a better choice of a template, I think that will be, if you kindly explain that
Yes.
thing.
See, between the two, these two are completely identical. So we can choose any one of this. If you look at here, also, these two are RSAR family proteins of Rhodobacter capsulatus. These two are also RSAR family proteins of Rhodobacter capsulatus, but here the E value is getting decreased. So between this set and this one, this is better depending on the E value. Since the percentage identity is the same, the score is little bit lower, but the E value is much lower. The E value is basically the reliability value. So these two are identical between this one and this one. So between this one and this one is better. Similarly, between these two, this one is better. So the basic thing is, first we need to identify the query coverage, the extent of the alignments. Then we would look into the identities. Then comes the E value. Okay, now this graphical summary, this gives you an idea about to which family the unknown protein. Here we consider the SOXR to be the unknown protein. The SOXR belongs to this family. This is the entire amino acid sequence of SOXR, and it is expressed in this way. This is the SOXR, and these are the results. If you do a mouse over, you will find the that means the amino acids or the proteins. This is the chain A, transcriptional regulative. This is also the chain A, and if we look here, this is the transcriptional activator of Vibrio vulnificus. So graphically, it represents the result is graphically represented. And if you look at the color scheme here, the alignment scores are in the range of this. Here, the alignment scores are in the range of this.
Yes, sir.
Yes. Any question? I think someone was asking a question, so you can ask. Hello?
Yes, sir. Sir,
Yes.
in the question answer section, no questions is there.
No question? Okay.
No, sir.
Actually, it's better to write because I can't hear anyone properly. Maybe this is due to the network issues. Now let us go into the alignment portion. This is basically the sequence alignment. See, the query means this is SOXR, the unknown, and the subject means this is the database sequence. From the 20th amino acid of query, the alignment started. And the 20th amino acid of SOXR, that means the query, matched with the 14th amino acid of the subject. This positive sign means identity or similarity. Positive, E. E means this is glutamic acid and D means aspartic acid. So similar amino acids are present at the positive. But F and A, these are dissimilar. F is phenylalanine having an aromatic ring. A is having a methyl ring. But this is exactly identical. So in a way, we can consider the positives to be the synonymous substitutions. This is identity, and these where no signs are present, these regions are non-synonymous substitutions. See the positives, you can count the positives here. You can count the identities, exact matches here. No gaps are present. This is the score, and this is the expectation value. The very high expectation value. Okay, I find one question. Oh, I find a question. Even though the score was this, how the percentage similarity was same. It may be it depends on the percentage similarity, depends on the portions that are aligned together. The scores are coming from the entire alignment. The percentage similarity depends. The percentage similarity depends on, say, among the 100 amino acids, there are 50% similarities. That means 50% identities are there. So that percentage similarity is 57%. And percentage similarity is basically the identity. By similarity here, we are pointing towards the identities, not the overall similarities. Okay. Now, these are the different alignments. These are the different alignments that you are having. You're having different alignments. And here, one gap. See, this is the gap. This is the region where there is a gap. In the query, there is a gap. In the subject, there is G in place of the gap. So we can consider this to be a deleterious mutation. An amino acid is deleted from the query, whereas in the subject, there is G. This is the gap. So, all of you got it?
Yes, sir.
All of you could understand the different points here?
Yes, sir.
Okay. So, see.
Dr. Bakshi, sorry.
Yes.
Dr. Bakshi?
Yes, yes,
Yes,
please.
yes. Sorry to interrupt
No
you.
problem. No problem.
Okay. So as you said that particular amino acid is deleted.
Yes.
Oh, in the query it is deleted, but actually that is the sequence of that particular organism.
Yes.
And it is being aligned with a known sequence which has a G. What I gather from your.
Yes.
So
yes.
the term deleted, I think, should not be used. Well, you can tell better whether we can say that that particular amino acid is not there. Is it
no,
true?
madam. No, it is not true.
How do we interpret?
It is not true. Here, what we are trying to do is we are trying to mimic the evolution. Our the BLAST tool it is also called an evolutionary algorithm. It is based on evolutionary algorithm. What it assumes is that the subject is the ancestor and query is its predecessor. That means whenever there is a gap, it considers that this particular amino acid G, which was supposed to be present in the query, is deleted from the query, and we are getting a new sequence on the basis of that deletion. This is the logic.
But suppose,
Dr.
Hello?
Bakshi, hello.
Yes.
Dr. Bakshi, if you allow me, can I add one line?
Yes, please.
Yeah, actually, what Dr. Bandopadhyay is asking, so yes, as per hard query, this is missing. Okay, but why missing? Because this is a new sequence. When the nature is on evolution is going, nature feels it is not actually important for this, so it is deleting from the sequence. Dr. Bakshi, may I write?
Yes, it is, yes, definitely.
So
that means
that's why this is the term deleted is used, because we are working with the natural sequences. Actually, this new sequence you are matching with another one. Okay, so what is finding this sequence is not found. What Dr. Bakshi is actually mentioned is very correctly. The subject one is our answer script, And one, if you are going for, suppose going for an OMR sheet, you find this is not there. So you consider from the aspect of the nature right, as
Yes,
in this way.
absolutely. Okay.
So,
Yeah.
and so suppose we have a query, we deposit a new sequence,
Mhm.
and then maybe nucleotide sequence, and we go for blast X, and we
Yes.
get the amino acid sequence and aligned, and
Yes.
we get this kind of, and that is a new sequence, and there is no error in the sequence, and that sequence is aligned with a subject, and there is a gap, and even then I can conclude what Professor Banerjee has said, that the new sequence which I have input into this platform is actually the sequence, and that G, if it has dropped, is because for some reason, because a living system will not keep anything which is
Not
redundant or not
required.
required, right?
Yes. Yes.
So
Absolutely.
with time, with evolution, it was found that that glycine was probably not required, and therefore is not required anymore, which was actually once upon a time existing. So am I correct? So this
And
is
nature
what the
has dropped the
interpretation, right?
sequence, and
Yes.
nature
Right.
has
So
dropped that glycine there.
exactly. So this is what we have gathered, right? Am I correct?
Yes, absolutely, madam.
Thank you.
Here,
Yeah.
here that means here a little bit of difference exists between that means the wet lab biologists and the computational biologists. That okay, deletion. That means the wet lab biologist. You people consider the deletion to be that you are mutating something. You have deleted an
Well,
amino acid, and
yeah,
you are
yeah.
trying to see its activities. So
No, not
from
always,
that
not
point of
always.
view,
What I understand,
from
not
that point of view, you are absolutely right that it is not deleted. It is the sequence of this. But from here, whatever Professor Raja Banerjee said or whatever you said, both of you are absolutely correct that this particular gene is dropped by evolution to get this new sequence. This new sequence might serve the same function as the older one, but the presence of G is not required for exerting its own function.
yes. Okay, okay. Thank you, thank you, thank you.
Yeah, there is another question, I think. What is the significance of plus? The significance of plus, as I told you, this is kind of the synonymous substitutions. For example, V substituted by I. These are the synonymous substitutions. Okay, so let us go back to the alignment parameters. Yes, there are certain things. I will explain the matrix, but let us start with gap cost. See, for gap costs, I told you that gap is kind of considered to be a kind of mutation, deleterious mutation. So while performing a sequence alignment, if you encounter a gap, that position, for example, in this particular case, there was a G in the query, in the subject, but that position, that corresponding position in the query is missing. It has no amino acid. So how to provide the score for that? Because all the matrices that I just showed you, all those matrices contain scores for alignments, but no scores for gaps. So that the score for the gap comes from here. That is called gap cost. There are two things, existence and extension. Here, in this particular case, you just find one gap. That is called gap existence. If there are multiple gaps, in that case, The first one is called the existence, and the next ones are called the extension of the gap. Is it clear now?
Yes, sir.
Okay, so you can change it as per your wish, and on the basis of that, you get different schemes, like different results. Now, these matrices, these numbers, there are PAM 30, PAM 70, PAM 250, PAM Blossom 80, Blossom 62, and all this. So, what do these numbers mean? Let us start with PAM. PAM, full form is percent accepted mutation. It means how much changes or mutations are allowed in a sequence alignment having proteins coming from the same family. Say, for example, we are aligning the amino acid sequences of the proteins belonging to a specific family. For example, say RHDH, DNA binding motif containing protein family. We are aligning all the members of this family. It is not possible that all of the members should have the identical amino acid sequences, but all of them belong to the same family. That means their sequence identities should be very high, but not all the amino acids are exactly identical. These numbers would represent how much mutations are there. That means PAM 30 matrix was built using the amino acid sequences of proteins belonging to the same family, but containing 3% mutations in them. PAM 70, 7% mutation. PAM 250, 25% mutations, and like that. So that way, these matrices were generated, and the basic idea of the matrices is to provide scores, right? So PAM 30 means we are getting the scores from this matrix, which was obtained from the alignments of the amino acid residues belonging to amino acid residues of proteins belonging to the same family, but they contain only 3% mutations in them, 3% changes. So considering all those proteins, we are getting certain values of the scores, the substitution scores. Similarly for the others, for the other matrices. So for highly identical proteins, you should start with PAM 30. And for dissimilar proteins, you should start with PAM 250. Now Blossom, the full form is block substitution matrix. The block substitution matrix, this matrix was built using the amino acid sequences of proteins which are not related to each other. And from there, the scores are obtained. So, in an identical way, the Blossom matrices were generated. But here, by default, Blossom 62 is found to be found to give a better results for the sequence alignments. These were actually obtained by performing many sequence alignments, by a trial and error method. So that is why the default values are, that means the default results are generated. Here, I need to mention one thing here, is that since PAM was obtained by aligning the amino acid sequences of highly similar proteins, so a lower value of PAM would reflect a higher, a better, that means alignment between proteins of belonging to similar families. But here Blossom 62 means this is the percentage of identities between the proteins which do not have any kind of family relationship. So higher values of Blossom would represent a kind of more similar proteins. Lower values of PAM would represent more identities, and higher values of Blossom would represent identities. But considering everything, Blossom 62 is found to give you a better result for all the types of alignments. This gives you an optimum result. But again, what I would like to mention here is that all these results are coming from multiple number of experimentations, multiple number of alignments.
Dr. Bakshi?
Yes.
Just one quick question.
Yes.
Since we are aligning all these similar and dissimilar proteins, what do you have to say about the alternative spliced forms of the genes? And the protein sequences could be a bit different or missing, but a larger part of the gene would have similar sequences. So in that case, if there are alternative spliced form, how are they considered in this kind of a search? Means,
yes.
Did you get my question?
Yeah, absolutely. So if you know that this is an alternative spliced variant, you know that, right? Beforehand.
Yes.
Okay. In that case, you can perform, you can use PAM, because you already know that this is an alternative splice variant. So there are certain variations, but these two belong to the same family. These two proteins, or these two, whatever, these two, I'm considering proteins only because proteins are mainly used. So these two proteins belong to the same family. You know the family information.
Yes.
In that case, you can use PAM.
Okay.
But a word of caution is that you have to be very much sure about it. Otherwise, your alignments would be meaningless.
I see. Okay. Yeah. We have to work it out. Then only we'll be able to understand how they are actually reflected through those alignments. Yes. Okay. We have to actually take these sort of examples and work it out ourselves. So, thank you. Thank you.
One more thing is that whatever the case may be, it is always better to first use the default value and then try to check the result. If you are getting results which are biologically meaningful, I would like to mention here is that we are entirely dependent on mathematics and statistics here. But mathematics and statistics do not understand the biology. This is a very, I'm telling you in a straightforward way. And we do not that means the mathematics and statistics do not understand the mechanism of biology, because in biology, not always 2 + 2 is not always 4, right? Because,
exactly.
for example, all of us have oncogenes. but not all of us have cancer.
Exactly.
Exactly. So that is why, madam, what I would do is I would first align align with default values. If I get a biologically result, then only I try to change the parameters, the default parameters.
Okay. So that means
answer
if
your
we are
question?
changing the matrix, like, this is the first time like I have been able to understand the usefulness of using a matrix. Otherwise, we always go for the non-redundant sequences and we directly go for blast. Either
Yes.
P or X or N, whichever we require, we just go for the non-redundant. And if it is like human sequence or any other Drosophila sequence, we try to make it more, the search more finer. But if we are changing the matrix, the scoring will change and maybe the result will be changing with the changing matrix, right? It will be reflected with the output, with the changing matrix, right?
Yes,
Yes.
absolutely.
Okay. Okay. Thank you. Thank you. You may go ahead.
Okay, thank you. Any other question from the participants? I can wait for
No, sir.
some time for the participants if you wish.
Okay, sir.
So, is there any question?
Till now, no, sir.
Okay. Now, here I would like to show you a couple of more things. That instead of using PDB, we can use SwissProt. In that case also, we will get some results. But let's see what results we are getting. See here, we are getting a different sets of results with different query coverages, e-values, and identities. If we go to the graphical summary, this would remain the same, but here we did not have this one in the previous case. And if we go to the alignments, if you remember, the first alignment in the previous case, it started from the amino acid 20, but here it started from 70. We get a different set of amino acids, different set of results. And all these results do not have any gap. Here, see, we get different sets of organisms as well. This is Vibrio cholerae, this is Agrobacterium, and so on. I find two questions. Okay. Sir, the query sequence is, query is the input sequence. Subject sequence is the database, is the sequence that is present in the database. Extension and existence, I mentioned a few minutes back that extension is the first gap. I'm sorry, existence is the first gap. And extension is the gap which is coming after the first gap. But what interesting thing that I was talking about is you are getting different organisms here. So what is important is that the database that you're choosing. In the previous case, we chose PDB. PDB did not have information of Vibrio cholerae or Agrobacterium fabrum. But SwissProt has it. That is why we are getting results, different sets of results from here. Why both gaps cost differently? Because always the first gap is the start of the mutation, and the second gap is the continuity of the mutation. That is the reason. Okay, then again go back and search it against RepSec. I showed you RepSec yesterday. See, here you are getting more and more of alignments, having these different values of alignments. See, you got so many. Here you have an extra where the score is greater than 200. See, these are the different alignments. You're getting so many different alignments. So, for the same protein, by changing the databases, you might get different results. That is the utility of these alignments. You can change the databases and you will get different results. Okay, any more questions here?
No, sir.
No more questions?
No, sir. There is all are you answered.
Okay, everybody has done it?
Yes, sir.
Okay, so this is the utility of sequence alignments. In a nutshell, what I can say, or what I can infer from here is that from the sequence alignment, we could identify the ancestor, or we could, I'm not saying I'm 100% correct, but we could predict the nature of the ancestor from the sequence alignments. Say here, we used SOX-I from the Pseudaminobacter salicylatoxidans. And in the results, when we used PDB, let us again do the search. See, here we are getting Rhodobacter capsulatus instead of Pseudaminobacter salicylatoxidans. We are getting a different organism. That means from here we could identify that, okay, Pseudaminobacter, salicylatoxidans, and Rhodobacter, these two might have some kind of relationship. ancestor predecessor relationship or something like that. That means we can have ideas about the phylogeny or phylogenetic relationships between these organisms. Okay, so let us again do another blast search. Say this one. SOX S. Let us take the FASTA. Search here. Let us first start with PD. Here, you are getting Paracoccus denitrificans, another organism. Let us do a quick search again with RepSec. Here, along with Pseudoaminobacter salicylate oxidants, you got aqua microbium, you got mesorhizobium. That means not all the proteins of the same operon would point towards the same or similar types of organisms. So the take-home message is you can sort the databases using sequences, amino acid sequences of proteins from the same organism. But it is not necessarily that you will get similar results always. There might be two reasons for that. The first reason is this SOX-R protein might be absent in certain organisms, or it might be the fact that the SOX-R sequence, the amino acid sequence of the SOX-R protein has not yet been sequenced from the other organisms. So these two might be the reasons for that. So I will now move on to my presentation. So before I move on to the presentation, I would like to have a couple of questions here so that I can carry out with the presentation. Any questions?
So till now, there is no questions in the question answer column or in the chat box. So we can wait for 1 to 2 minutes. Then
Okay, I find three questions. One is from Momita Shaha. All the three are from Momita Shaha. So according to then whatever database and mentioned mention them while debriefing? Absolutely. While writing the paper, you should mention the parameters that you are using. And this question, we have to practice using different databases and other aspects to get the better result. Absolutely. For a sequencing experiment, don't you have to perform multiple times? This is also an experimental biology. Computational biology is nothing but you are sitting in front of the computer and getting the results. It's not like that. You have to perform experimentations. I gave you the answer of this question. You have to mention the databases and the matrices that you're using, because when you are writing a paper, you're providing your results. The referee who is going to check your paper would like to have your data checked before passing a comment. So you should clearly mention that.
Two more questions are there.
Yes, significance of 1E. E stands for 10, 1 into 10. 3 is 3 into 10. That is E. So here you can see it is 6 into 10 to the power minus 83. That is the E value. Suppose we are getting better alignment result using some different parameters, but in that case, there is a gap. And while going with other databases, we are getting good results compared to the previous. I don't understand. I can't understand your question. We are getting better results. Better with respect to what? Better alignment result using some different parameters. Better with respect to what? Better should be respect to something. While going with other databases. Second one means in case if we choose a protein that has been sequenced from other organisms, you get similar results by choosing. Sequenced from C. What the thing is that you got the amino acid sequence of a protein from some organism. If you know the ancestral history of that organism, you can then search using that ancestral history, using that information. For example, proteobacteria. If you know your organism belongs to alpha, beta, or gamma, or delta proteobacteria family, say your organism belongs to delta proteobacteria family, and you sequenced the amino acid sequence of a protein from that organism belonging to the delta proteobacteria family. Belonging to delta proteobacteria family, right? Now, what you can do is, you can, while searching, you can use the database which stores the information pertaining to delta proteobacteria family organisms belonging to delta proteobacteria family. That you can do. But if you don't know anything about the ancestor, in that case, you should either choose RepSec or SwissProt. Your question is not very clear to me. Second database means what? can you please tell me if the blast result for any particular organism remains same? Yes. They may not remain the same, they may remain the same. Generally, in Swiss prot, the sequences are present. But in PDB, both the sequences and structures are present, but only those sequences you will find in PDB for which structures are available. So it may so happen that you have only the sequence of an organism, but that sequence has not yet been converted to any structure. That means the structure from that structure of that particular protein has not yet been obtained. In that case, that sequence information will not be present in PDB. In that case, you might miss it. Second parameter gives not like as much as good alignment like the first parameter, but in case of alignment with second database, we are not. Your question is still not clear. Anyway, what I could gather from your question is that you are having an amino acid sequence of a protein and you are trying to analyze that sequence. Trying to analyze that sequence from two different databases. But while considering the parameters, I told you that not to change the parameters. You can change the parameters one by one. First, you use one set of parameter to search in PDB and Swiss prot. Then you use the second set of parameter to search PDB and Swiss prot. But generally, the default parameters will give you the best results. Sequence alignment results enough to establish phylogeny. I did not mention that. I did not say that it is enough to establish phylogeny. I say it is a prediction. It gives you a prediction towards the ancestral history. Okay, any more questions? Any more questions?
No, sir, all you answered.
Okay. So let us then move on to this one. And this is dynamic programming algorithm. And this is dynamic programming algorithm. Probably this requires much statistics and mathematics. And from the point of view of analysis, this is not so important. So I'm just brushing through this. Here in dynamic programming algorithm, number 1 is given where there is an exact match, A with A, 1, Y with Y, 1. And that way the alignment is calculated. So see here, this is an important one. This is called the trace back. So from the last portion, we would like to trace back. So P aligned with P, and then it moves like this, and this way the alignment is done. So see, this is the alignment, P with P, then comes B aligned with nothing, and then R aligned with R. since it got 2. C aligned with C, K aligned with L, and that way. So there are multiple such of trace packing, and all these will give you different results, different alignment forms, and different scores. Now, this one, global and local alignments. Global alignment means alignment of the entire sequence. Alignment of the entire sequence is called the global alignment. And local alignment means alignment between a specific stretch of a sequence. This is the local alignment. And here, gap penalty, open and extension. The first gap, this one, these two are called the gap open, and these are the gap extensions. I mentioned this. Now I am moving on to multiple sequence alignment. And so let me give you the background, the basic of multiple sequence alignment. So what is multiple sequence alignment? I told you earlier that multiple sequence alignment is the alignment between more than 2 sequences. So the basic logic is what we have to do first. We have to first perform pairwise alignment. From pairwise alignment, what we could gather is the amino acid sequences which are identical. That means we can have, say, you have an unknown amino acid sequence of a protein. You just have the amino acid sequence and have no information about it. Now, what you are doing? You are trying to identify its homologous sequences. You perform a pairwise alignment. After performing a pairwise alignment, what you could identify is, you could identify its homologous sequences from the databases that are present. Now, what you would like to know is, what is the relationship between all these homologous sequences with the sequence that you have? For that, we perform multiple sequence alignment. So the thumb rule is, first we have to perform the pairwise alignment. Then, after getting results from the pairwise alignments, we perform multiple sequence alignments between the results that we obtain from pairwise alignment. So there are several methods of multiple sequence alignments. So this is kind of pairwise alignment, pairwise comparison. And what we do is, this is pairwise comparison between A and B, A and C, A and B. Now we perform alignments between A, B, C, and D together. This alignment, we will do the hands-on portion right now, but before that, mainly these are the important ones, but now cluster W is substituted by cluster omega or cluster X. Okay, so let us again do the pairwise. Okay, let us do one thing. Let us go back here and this is the result of SOX protein, right? So let us have the protein from here, SOX. Let us have its faster sequence. Let us copy. And then let us type cluster omega, which is a tool for multiple sequence alignment. Here, first, let us paste it. Here, a little bit, one thing you need to perform, you need to have a continuous sequence. From here, let us consider this transcriptional regulator. Okay, participants, all of you are doing this. All of you can follow, right? Or am I moving too fast? Participants?
Sir, I think it will be better if you go a bit slow.
Okay, okay. So let us start from the beginning then. Copy and paste the amino acid sequence of success here. Then come to blast result. Let us choose this one. Click here. We get this. Select the FASTA from here. Select it and paste it here. Make it continuous. Okay, I'm waiting. Let everybody do it.
So there is a question.
Oh,
Can sir repeat the cluster omega portion?
cluster omega. Okay. The cluster omega is okay. Just type cluster omega in Google. Once you get this, select it. Paste your sequences here. Let everybody do it, then I will continue. Okay. Then again,
Sir, I think it will. Everybody can done in this time.
okay. Then again, and choose the other one. We need to have at least a couple of sequences in order to do the multiple sequence alignment. Go to FASTA. do the same stuff. Select it, paste it here, make it continuous. Then again do the same thing. Choose this one. So let us stop selecting here. Tell me once everybody is done. Tell me once everybody is done, we'll continue.
Sure, sir. We'll just, thanks, sir. I think we just need to wait 1 minute more, then we can proceed.
Yeah, no problem, no problem, no problem.
Okay, sir.
Sir, you may proceed.
Okay.
Yes,
So,
sir.
okay, now do a submit. See, this is the multiple sequence alignment. These are the sequences. These are the results. See here, these stars would represent exact matches here. The double dots, these double dots represent the synonymous substitutions. And these are the numbers. That means it starts from 1 and the first line ends at amino acid 59. This is the first amino acid of the second sequence. It ends in 54 and that way. These are the regions where no matching has been found. So from here, what we could gather is there are a couple of portions like this portion, this portion, this portion, this portion. These portions are exactly identical in all the amino acid sequences or all the proteins. So we can consider these portions to have certain relationships or certain functional relationships, I should say. For example, these portions might be considered to be the active sites of the proteins, though this is a prediction. I'm not saying this is 100% correct, but in an indirect way, we can say that if we know the active site of one particular protein, and if we can perform a sequence alignment of that protein with the other members, that means other proteins or other members, where the active site region of the known protein matches with the other members, in that those regions might be considered to be the active site portions of the other proteins. So from multiple sequence alignment, we could have an idea about the possible functionality of the protein or the conservedness, amino acid conservedness of the protein. There are certain options like show color. It makes the alignment highly colorful. It looks easy. If you check the results summary, from the result summary, you can have several things. These are the input sequences that we have given as input. Let us see the tool out. The most important thing is that this gives you the CPU time. That means how much time it needed to compute the entire process. This is the result. This can be used for publications. And this one is important. See, this is the percent identity matrix. This one has 67% similarity with this one. This one has 61% similarity with number 3. One has 66% similarity with number 4. So this is called the percent identity matrix. And you can draw a phylogenetic tree from here. This is the phylogenetic tree. The most similar ones, these are the most similar ones. These are the most similar ones, and this is an outgroup. That means these two are clustered together, these two are clustered together, these two are clustered together. So these two and these two, they are related by this. This entire one and this entire one, they are related to this in this manner. So this is a very crude phylogenetic tree, but this is that is a kind of phylogenetic tree that you can obtain. That means at least you can have an idea about the phylogenetic relationships. I find one question here. Oh, single dot means synonymous substitution. Non-synonymous substitution. Sorry, double dot is synonymous substitution. Everybody got the results?
Yes, sir.
And the phylogenetic tree. So it is very simple. If you click here, you get the phylogenetic relationships. So in the phylogenetic tree, these two sequences are clustered together. They are similar. These two sequences are clustered together. So they are similar. Together, these four sequences are similar since they are present on the same side of the tree. These two sequences are also similar. They are present, they are different from these two. That is why they are present in a separate plan. And this one is an outgroup, which is kind of associated with all of this. Okay. Yes, everybody got the results?
Yes, sir.
Okay. So, this is a way to analyze the sequences belonging to the same family. I told you that you have to perform that means multiple sequence alignments after performing pairwise alignments, because see, in the process what we did is we took the results from the blast search. Blast is always giving you the pairwise alignments. So here we used this sequence SOX S. We aligned the SOX S with the sequences that are present in the databases with the help of BLAST. The results that we received from BLAST, the basic idea is from the results, we could gather to which family the SOX S protein belongs. So we picked up all the existing family members to which the SOX S protein belongs, and then tried to compare their amino acid sequences. That we did with the help of multiple sequence alignment. We got the results. Let us show color and see these portions are completely conserved, absolutely conserved in all the sequences. So these portions might be considered to have certain functional or structural roles. Conserved portions. The double dots are the synonymous substitutions. See, valine leucine. And the single dots are kind of non-synonymous substitutions, like glycine T aspartic. And this is variable, where nothing is present. See AR, that means alanine and arginine, not similar at all. Any more questions you have here?
Dr. Bakchi?
Yes.
Yes. Can you go to the next slide in that phylogenetic tree?
Okay.
The cladograph, yeah, exactly. So I just want to know, see, you have the top two clustered together, followed by the other two clustered together. And there's a bottom one also, they're clustered together, and there's one intermediate one. All
Yes.
of them, they belong to the same family, but there are sequence similarity and they have been clustered together, what I understand, right? Because the
Yes.
branching point indicates that they belong to the same family with variations.
Right.
So my question is, when this clusters are made, like the top one versus the immediate, the first, second, and the third, fourth, means from the top,
Yes.
how much percentage of dissimilarities would break them into two different clusters? Means this is my question. Or the other means there are similarities, but how much they endure or they take in to keep them in a cluster, and how much it is allowed, and then they will be separated. That much. Maybe you have mentioned it. I missed it out. If you could
No,
explain
no,
once.
yeah. So if you go to the this portion, I think result summary,
Get my question right?
and this person identity matrix. So from here, see the first one has
Yes.
the maximum similarity with the last one, 71.8. So these two are clustered together.
Okay.
So here it is given. So yeah,
So
I
from
got
this,
the point that is there any cutoff? So no, there are no
Yes.
specific or predefined cutoff. It depends on the alignment score.
okay. So this is actually whatever we search, it will be automatically reoriented from this particular platform. Means so from this tree analysis, right? The program will take care of it. The program will take care of it, right?
Yeah, the programming will take care of it. Absolutely.
Okay. Thank you.
Any other question?
No, sir.
No, sir.
Okay. So from here, what we could gather is that we can have a fair amount of idea about the similarities between the different types of sequences. So if I go back, see, these are the algorithmic details that we need not have to do it. This is the blast that is performed here. The things that we need to remember a little bit, that is local. BLAST stands for local alignment. That means BLAST is not global alignment, as the name implies. And there is this maximum segment pair. Maximum scoring segment pair means it that entire sequence is broken down into pieces, and BLAST tries to match that piece with the other sequences that are present in the database. So that way the alignment is done. So BLAST is the basic local alignment search tool. So this we performed, we did it in the hands-on portion. So let me give you some ideas about the gene finding as well. So that is another important portion. I'm sorry. Yes. Gene finding. See, this is all of you know, this is the central dogma, and all of you know it very well, how the chromosomes are generated. And so what is gene finding? Gene finding means, and this is a part of this sequence alignment technique. This gene finding method, so far we considered only the protein sequence, amino acid sequences of proteins. This gene finding method is used to analyze the nucleotides for all these reasons. These are the completely sequenced genomes of all these organisms. And the Human Genome Project, we all know that 99% of human genome has been sequenced so far. And a new concept has come up. Okay, let me have the question. No, not any cutoff. Okay, so one thing I must mention here is that the Human Genome Project, from the Human Genome Project, we could identify, or we could gather the information of some long non-coding RNAs. Those are the non-coding RNAs which do not code for any proteins, but they have very important roles in disease development. So we, till now, we do not have very good ideas about those long non-coding RNAs. And so gene finding would help us to identify where the actual gene is, to which portion. That means from the entire genome, if we could identify the genomic portion, that means the genes and the gene region, the remaining portions are considered to be the non-coding portions. So what are the effects of those non-coding portions? That is very vital. And there are certain methods with the help of BLAST, we could identify, with the help of multiple sequence alignments, we could identify in a similar way for proteins. It is mentioned here, BLAST. And there are two things, intragenomic and intergenomic. This is applicable. I told you in case of proteins that the proteins belonging to the same family and proteins which are scattered, which do not belong to the same family. It is, the concept is the same for this intragenomic and intergenomic species. And this is called the ab initio method. So, but the complication is much less here, because in case of nucleotides, we only have 4 letters, A, T, G, and C. But for proteins, we have 20 different amino acids, and there are multiple combinations. So more varieties are present in case of proteins, but in case of nucleotides, much less varieties are present. So that is why all these sequence alignments, we focus mainly on proteins. So I'm not going to the details of this. Yeah, and this one is a new method which is different from BLAST. and multiple sequence alignment. This is called the machine learning based approach. Here, what we do is we extract certain features of gene, certain sequence features, and we train a machine learning program with those features. We feed them the positive features, that means features of the gene, and the negative features, that means features which a gene should not possess. And we feed them to a program, a machine learning program. The machine learning program, on the basis of that knowledge, would come up with a logic that if an unknown sequence is given as input to that program, machine learning program, it could identify whether the unknown sequence is a coding sequence or is a non-coding sequence on the basis of those features. The features are collected from the genomic regions of organisms that are present, that are already established. So it is easy to get the positive features, but it is difficult to get the negative results because, as I told you earlier, that in case of nucleotides, the variations are much less. So, since the variations are much less, it is not, there is no clear-cut demarcation between a gene and a non-gene part. However, machine learnings are found, that means are constantly giving better results, but there is a word of caution. The machine learning results are heavily dependent on the training data. A good training data set would provide a better result than not so good training data set. And a good training data set should have a balanced collection of positive and negative examples. And then there is this cross-validation. Cross-validation means the result, the machine learning result is run for several iterations. And if we get the similar results in a majority of the cases, we can consider that result to be reliable. But if that result varies, in that case, we need to perform more and more experimentations. So that is called the cross-validation of the data. This is, I told you that it depends on good training. And these are certain tools for gene finders. And all these tools are basically dependent on machine learning results, like the neural network, like the support vector machine, like the Markov models, and so on. So this is another aspect of sequence comparison, but this is basically, and this sequence comparison is basically to identify genes, and the gene identification is coming directly from the genomic sequence, that means the nucleotides, not the proteins. Now, the next thing is, can it not be used for amino acid sequences? Yes, this method can be used for amino acid sequences as well, but that is problem specific. For example, if you want to know the presence of a specific motif in a particular amino acid sequence, or if you want to know the family information of an unknown protein, you can use the machine learning method. In that case, let us take an example, say thioredoxine protein. So from the amino acid sequences of all the thioredoxine proteins, some features may be collected, which would give you an idea about the characteristics of thioredoxine proteins. This is the positive set. As a negative set, you can use the features from any protein which do not belong to the thyroidoxin class. And with the help of those features, you can train a machine learning algorithm, and then the machine learning algorithm, the machine can discriminate between the thyroidoxin and the non-thyroidoxin family of proteins. But again, for proteins, it is very much family specific. This is also entirely, all these are also entirely dependent on sequence comparisons, the multiple sequence alignments, because from multiple sequence alignments only, you could identify the regions which are conserved, and from the conserved regions, we could identify the specific amino acids, the presence of specific amino acids, and the natures of those amino acids. So this is another aspect, which is an upcoming aspect of sequence comparison. So with the help of sequence comparison, what we could do is we could identify the family information, number one. We could identify the possible structure, because if we could identify the family information, from the family information, we could identify the presence of the distribution of secondary structural elements. From the distribution of the secondary structural elements, we could definitely identify the pattern of the two-dimensional structure of the protein. From sequence alignment, we could identify the conserved regions. From the conserved regions, we could identify the specific features of its functionality. So all these important aspects may be considered or may be obtained from sequence alignment technology and methods or techniques. Thus, this gene finding methods, or this categorization, or the discrimination, or the distinction between the family members, those can be done with the help of support vector machines or any of the neural networks, genetic algorithms, or all these machine learning based techniques. And all those machine learning based techniques are also dependent on the sequence comparison. So that is the utility, that is the beauty of sequence alignment. Let us see what questions are there in the chat boxes. Yes, so so far this is all about the sequence comparison methods and how you will perform the sequence comparisons. If you have any questions, we can discuss the questions. And also for this is that after the pandemic is over, in Kalyani University, we have a bioinformatics center, and we'll have workshops. We'll conduct workshops from the bioinformatics centers on different aspects of bioinformatics. So I will send you the flyer for that. And also we are open to collaborations. If you have any specific problem, we can try to help you out for using our resources. So over to all of you. If you have any questions, queries, we can have them here. See the first question. You can use all these. The names are present here.
Dr. Bakshi?
Yes.
Yeah. I have one question. When you mentioned that gene finding, the tool, right? What is the full name? I just missed. Gene finding.
Yes.
So I have a question over here, that when when you are talking about when you are actually pinpointing a gene, then that gene should start with a promoter with all the genomic sequences of exon, introns, everything, and ends with a trailing sequence, and that's a complete gene information. Especially in the higher, in the eukaryotic system, they will have these complex sequences in between. But for the prokaryotes, this is much simpler.
Yes.
So every gene will be starting with a promoter and then the downstream sequences and the complete gene. Now, my question is, will that gene finder or that particular tool uh, uh, demarcate? Suppose you give a long string of sequences, say like 50 kb sequences. In that 50 kb sequences, you have maybe 7 genes. This is for eukaryotic system, if I am thinking. So 50 kb sequence you have input into the gene finder. Will it be able to pinpoint with color codes or something of that sort, and indicating that from the top strands there will be 7 genes, and from the bottom strand, which can also be transcribed in the reverse direction if the promoter is in the 5' end of the bottom strand, that the number of genes existing into that 50 kb string of sequences, if whether that gene finding tool can help me to locate the start and the end of the gene.
Okay.
Will it be possible?
I'm giving you a question. Answer to your question in bits and pieces. First, for the individual elements like the promoter, like the enhancer, like the transcription start site, for all these, to identify all these regions, there are specific tools for that. So you need to first give your sequence to a gene finder. The gene finder will first give you the results of the promoters, of the enhancers, of all those sequences. The things that the amino, not the amino, the nucleotide sequence from where the first promoter starts and where the first promoter ends, and then the enhancer, and then that means the transcription start site and the transcription end site. All these. So it does for both the 5' 3' and 3' 5' directions. And it gives you some probability scores of all its results. The result having the maximum probability score might be considered to be the most reliable one.
Thank you. So that means if I narrow down my search and I take a 10 kb genomic fragment, and
Yes.
it is having, say, 2 small genes, means roughly about 4 kb, the genomic sequence of the individual genes are, say, about 4 kb. And I have input, I have been successful in getting the entire 10 kb fragment, which is housing 2 individual genes. So your gene finder will be able to pinpoint those 2 genes start and end through some calculation, and it is going to give the output, right? That's what you mentioned. If I
Yes.
get, if I'm not wrong, right?
Yeah.
One
That's
more.
true.
So that, okay, and then so this is for one strand, say the top strand, which is 5.3' directed, and there is this bottom strand which also can have other 2 genes, or maybe 3 genes, or maybe only 1 gene or no gene, because the promoter is directed in the opposite direction, and it is going to have a separate information, not it is only a complementary strand of the top strand, but it will have a totally different information with respect to a gene. So your gene finder will address both the strands, right? The reverse and the forward.
It will address.
Okay.
But here,
Okay.
madam,
Yes.
like as I told you earlier, that all these results, you have to scrutinize the results with your experimental findings. Like say a gene finder and gives you a result of say with a probability score of 0.9. That means 90% probability that this is the transcriptional start site. Okay, but as I told you earlier, that these gene finders are based on certain existing data, that is features that are collected from the known genes, the transcription start sites of the known genes. It might be a case that, as I told you, that this is a statistics and mathematics, but biology is a little bit different. So it might be that that transcriptional start site might have certain different features. So I always request everyone to compare the results that are coming from bioinformatics with with your experimental results. But along with that, I would also like to say that this would also help you streamline your experimentation. Like you can target that particular region to identify the gene instead of targeting everywhere.
So that means the gene finder is very useful for those genes which have been identified. Okay, because
Yeah.
you get because they are already reported, and it has been even the predicted sequence is also available. Might be that the experimental part is still not known, but some prediction through computation is always there, which
Is
has to be
always there.
revalidated, with the real experiment that you are always emphasizing, that biological data might not have the same results from the predicted through the computation. That's absolutely well taken. So which
Yeah.
means, what I understand, for novel genes where you don't have any information, I think doing a blast and having that roadmap, and then maybe we can input that novel sequence into a gene finder. Is it rational? Means, is it okay to put
Yes,
input?
it is okay.
Is it okay? It is
Yeah,
okay,
it
right?
is okay.
Okay. Because at least the program will take care of the start of the gene, the end of the gene, by some sequence homology, or some basis should be there, because a promoter is a promoter with all the sequences which are conserved. The
Yeah.
consensus will actually define a promoter after all. So novel genes can be used into the gene finder.
Exactly. And one more thing here I would like to mention, that all these algorithms, that means the tools, which are based on both structure and sequence information, those tools are much more accurate than the tools which are based only on sequence. The tools which were developed using the sequence and structure features, those tools can give you accuracy up till 90 to 95%. But definitely, you have to have sequence and structural information together.
Thank you so much.
Any other question? Any other questions?
Sir, I can't see any other questions in the question answer box and also in the chat box. So
There is no question.
I don't find any question in the question answer box.
yes, sir.
In the chat box, let's see. No. Okay. So as I told you, that we have a bioinformatics center. So if you require any help in performing your bioinformatics experiments, so you can contact us. We'll try to help you out. But definitely this pandemic should be over, because the university is closed till 30th of September. And so let's see, hope everything will be fine after this pandemic. And we will have fruitful collaborations with your university, since we are very near to each other. We are kind of neighbors. So we have collaborations with ISER and NIBMG. So it would be great if we could have a fruitful collaboration, if we could construct or make a fruitful collaborations with Maulana Azad University as well. Thank you very much, everybody. And I request Dr. Poti and Professor Raja Banerjee to kindly provide me the feedbacks of the participants. And thank you.
Thank you very much, sir, Dr. Bakshi, for your restless and wonderful session. I believe it has been a very useful session to all the participants and researchers. Students have learned about what exactly the sequence analysis, like pairwise sequence analysis, multiple sequence alignment. Also, we learned the clear view of dynamic programming, and it is widely used in bioinformatics for sequence alignment, protein folding, RNA structure prediction, etc. Dr. Bakshi has covered very deeply BLAST algorithm for comparing primary biological sequence information, such as amino acid sequences of proteins or the nucleotides of DNA, RNA sequence, and compare a subject protein or nucleotide protein with a database of sequence and identify database sequences. Also, he covered usefulness of BLOSUM and PAM matrices. Hope all the participants taking up this subject should understand. Dr. Bakshi has covered interestingly BLAST for Swissport, Uniport data, etc., and explained very well. He also learned about the concept of clustal omega, a multiple sequence alignment program, and how it is produced biologically meaningful multiple sequence alignment of protein-protein sequences using phylogenetic data structure, color codes, etc., by picking up certain examples for the students to better understand. He clearly described about gene finding in terms of intragenomic and intergenomic concept. Also, we understand about how machine learning program works on biological data with some intrinsic parameters with an example of amino acid. So thank you very much, Dr. Bakshi. It has been very interactive session through question and answers put forward by our participants, and respected panelists cleared some basic doubts which help all the understand better. Thank you very much, Dr. Bakshi, for your wonderful session. Now, I request Professor Banerjee to share his valuable views on today's session before the audience and give a vote of thanks to our respected speaker, Dr. Bakshi, and the participants. Professor Banerjee, please.
Before I talk, I will rather request one of our participant and our faculty member, Dr. Joya Bandyopadhyay, to say a few words, and then I will talk.
Okay,
Dr. Bandyopadhyay.
Okay. Thank you, Professor Banerjee and Dr. Poti. And definitely I thank Dr. Anshuman Bakshi for this wonderful workshop. And why I have enrolled myself as a participant, I am not supposed to be here because I am a participant today, not as a panelist. Nonetheless, I feel honored that I am one of the panelists. I just want to share one experience from this this 2-day workshop. That is, this is not the end actually. This is just the tip of the iceberg, as our Honorable Vice Chancellor pointed out in the morning, because it is just a part of some sequence information, DNA, RNA, and you have elaborated on the protein, what the protein sequence today mostly. It is just the tip of the iceberg, and there is much to learn because the biological system is a mystery. And whatever is predicted through all these different software and all these computational platforms um, is just a prediction, very much useful, and of course uh, has to be validated with the real biological data. And it is going on, research is going on, and there are actually, we get excellent publication. And the students should know that when both the computational and the biological aspects merge, or if there is a marriage between the two, then the outcome is an excellent publication. So only prediction is a first step, and then the rest lies on the biological data, and the two have to be collated together. What I have understood also, that we are just the end user. Dr. Bakchi, we are actually not so sound on the programs, on the different computer languages, because we are just the end user. And from the point of the end user, sometimes programming gets a little bit heavier for those who are not computer professionals. I think you will agree on it. So there might be certain results or outcomes of the data which might not be so easy to understand, but at the end, what we are looking into is what is the sequence going to speak for. So we are the end user, and from the endpoint, and from the point of view of an end user, I have been benefited from your lecture. But yes, more such sessions have to be conducted, and there are many more topics that can be covered in such workshops with the real hands-on that you have actually made us work for yesterday and today. So for the students, this is definitely very, very helpful, because our the topics that we cover in the class has got much to do with what you have covered in these 2 days. It's very, very useful. It's going to be useful, and it will make our work easier when we go for these type of topics about gene, expression, and sequence. And it will make my job simpler in the days to come. And I really congratulate you that you have done an excellent job. And I also thank the bioinformatics department to take this initiative to arrange this kind of a training session. This is a true training session, and we could concentrate more, because all of us are hooked up with this laptop. So we got a very one-to-one one-to-one train knowledge. So thank you, Professor Banerjee, that you have actually made this workshop open to all, and open to me also as a faculty. I am a participant today, and I'm very thankful to the entire team. So thank you very much once again for giving me this opportunity to share my views. And I hope students have benefited, and there will be more such sessions, and we will definitely request all the students to post their questions even after this workshop gets over, to our respected resource person, Dr. Umshuman Bakchi. He will be, I think, very happy to answer all these questions. Your mailbox might be flooded with all these questions in the days to come. Thank you so much. Thank you.
Thank you, Dr. Bandyopadhyay. Actually, I am finding Shoham Banerjee. I think he is a student of our just who passed the degree, and who is also actually acting as a back person or the back team. So may I request Shoham to say a few words on behalf of the participant, because he has an idea about he has attended few classes. Now he has seen a after having some experience in the school class or school. Now he is showing what is the research area. So if Shoham say a few words, then I will say, or if any other participant is there who can, because this is a very, very interactive session. I'm very happy. So Shoham, one, and if you can arrange one or two participants to say a few words, so I think that will be very good, great thing to us.
Thank you so much, sir. I'm really very much honored today by your cordial words. At first, I want to thank our Honorable Vice Chancellor, sir, for giving us this kind of opportunity to arrange this kind of informative and innovative session. After that, I want to thank Dr. Shoman Poti, sir. He requested me to participate, and also he gave me the opportunity to be a panelist here too. And I don't think I am that much capable to be that panelist, but I'm really cordially appreciate you guys' efforts and this respect. And after that, I want to thank Dr. Anshuman Bagchi sir for his kind time and this cordial responses to each and every answers, which are very innovative and informative for all of us. Because bioinformatics is still now a very informative session for everyone. Not everyone is capable to understand this kind of high data analysis and sequencing parts. So this kind of workshops quite helpful for us to deal with this kind of things. So up to this, that's all, sir. And now I would like to request some participants to please kindly share their viewpoints about it, and hope we'll get many more such kind of workshops in near future. Thank you, sir. Thank you so much.
Yeah, Shoham as a back person, yeah, Shoman, tell. Shoman, if you want to say something.
No, but you carry on, sir.
So Shoham, can you arrange one or two participant, as Dr. Bakshi will be happy to know what is the thing that's going here.
Yeah.
Sure, sir. I will try. I will try my level best. Just give me 1 or 2 minutes, sir. So I would like to request Prem Bhaiya, please unmute Shouris Bhattacharya. He is from BTech Biotech from UEM. He would like to share his thoughts about this
And
informative
another
webinar.
one is Momita Saha. Another one is Momita Saha.
Yes, sir. Sir, sorry, sir. Bhattacharya, sir, please, you may ask the question.
Hello, am I audible?
Yes, sir.
Yes, you are.
Please share your experience, sir. Please.
Yes, good morning to everyone. Actually, it's now good afternoon. So it's really great to be a part as a participant in this workshop. It's a really very enlightening one. And thank you for presenting this type of workshop in front of us. It's really very knowledgeable. Thank you. That's all.
Okay, thank you too also.
Sir,
Another
now
one,
we may request Momita Saha to please say something about this program. Please, Momita Saha, unmute your mic.
Hello.
Yes,
Hello.
please.
Good afternoon. My name is Momita Shah. I am Tech Media University right now. Well, in this, I would like to say that this is a very wonderful. I think today's session is more interacting and wonderful to attend and gain knowledge about different cluster W's software tools, their plus Blast, etc. So thank you for MAKAUT for arranging such a wonderful session on bioinformatics, because right now in today's pandemic situation, we can't go to the lab and do the experiments hands-on. So if we get a chance to know more about the bioinformatics, then it will be easier for us to do some dry lab sessions in our own home. So thank you, MAKAUT, for arranging this kind of workshops. And I would like to tell one more thing, that if in future, if possible, arrange this kind of workshops more, so that we can attend these seminars too. Thank you. Thank you.
Okay, thank you.
Showman, can I speak one more time, if I'm allowed?
Yes.
Professor Banerjee, am I
Yes.
allowed?
Yeah, sure.
Yes, actually, this definitely this workshop was intended towards the sequence and the databases, because obviously we cannot cover, or anybody cannot cover in the 2 day so many topics. Bioinformatics is a huge subject, and there are so many facets that we all need to understand. Even learning everything, we will not be a true bioinformatician, definitely not. We can be an expert in a very small fragment of this subject. I would request the bioinformatics department to hold more such workshops and training. And this next time, maybe docking and protein interaction, and more towards drug and receptor docking, these sort of workshops and training will be also useful. It is just my suggestion to the department, because since you are going to plan more for the near future, kindly arrange at least 1 or 2 day workshop uh, where you can teach us how we can manage these docking. So this was my suggestion only. It is just my view, and I hope it will be one day. This would be addressed one day. Thank you.
Thank you, madam.
So
Now,
last
Professor
but not
Banerjee.
last but not least, so this is the thing. Actually, what I have to say already it is said by Dr. Bandopadhyay, Dr. Mr. Shoham, and Moumita Shaha. So I am really thankful to Dr. Bakchi for giving us his valuable time from his despite his busy schedule. And actually, this is a joint effort, jointly organized as a coordinator by Dr. Poti and myself. But I am actually very humbly accepting this thing. The full credit, 100% credit, goes to Shobhan Poti, who is basically organizing the whole thing. I have not done anything. If anything, any responsibility, I think as a senior it comes on mine. But the whole credit goes to Dr. Poti for his for making this workshop as a grand success. So first of all, I want to thank Dr. Poti for making this as a grand success. Secondly, I also thank to Dr. Bandopadhyay for sharing her views and also suggesting to organize. Already we have in the last June or July, I think, Dr. Poti, we have organized a 5 days workshop in July.
Yeah.
Where we have already
June.
June. Already we have discussed or you have arranged this thing, how the force field has come, what is the requirement of the force field, and how this can be applied. Actually, as you are suggesting, we will think about that. And last but not least, I must thank to Dr. Bakchi once again for giving us precious time, giving us, actually this is not a thing, this is not a simple thing, this is basically a ocean. And this is from the ocean, he is giving you just 2 or 3 buckets, but what he has given, that is very helpful if you understand properly, and if you want to cultivate by yourself. And the self study is the best study. If you go, I think Dr. Bakchi is every time he is, if you can send some question, he will be happy to help you. And already he has given you an opportunity that actually we have also in our university there is a center, and he has also has a center. So we can plan together also, because both are very close to each other. So once again, thanks a lot. Thanks also to our honorable vice chancellor for allowing us to do this thing. So thank you once again for your cordial participation. This is for the participants. If you are not here, if you are not interacting, actually this session will not be very so uh, good. So once again, I am very happy to say this is a very, although it is an online, but the interactive session is very effective. Even not this interaction, what I found, it is even not found in a real session also. So Dr. Bakchi has answered, tried to answer all the questions, and I'm also grateful to him. He's allowed me also to tell in between him also 1 or 2 words. So thank you so much, dear participants. We are looking for you once again, and on behalf of the WBUT, now MAKAUT, and Department of Bioinformatics and School of Biological Science, once again, very good afternoon and have a good day.
Thank you so very much, sir. Now, Prem Kumar, is there any announcement for the participant?
Sir,
Please,
we
you-
will be providing the e-certificate after the quiz and feedback.
Okay.
So shall I share the links?
Yeah.
Yes, sir. There will be a quiz, sir. The quiz time will be half an hour. And after that, giving the quiz and feedback, the certificates will be available to download after 2 hours, sir. So the links has
Is the
been
links
posted.
visible?
Yes, yes, yes.
So all the participants, you please take a screenshot of this, please. So thank you, Prem Kumar. So the participant, you please fill up the feedback form with some multiple choice questions for the limited outcome of today's session. This is mandatory for all the participants. If you do that, then you can get the certificate. So hope you enjoyed the workshop with the hands-on training and benefited with both these sessions. Thank you again to all for joining the workshop. Thank you all participants. Hope we'll meet again. Already time is over, Prem Kumar. So
Yes, sir. Yes, sir.
we may close.
Yes, sir. So for 1-2 minutes, let the links be available so that they can note the links. Yes, sir. It is requested to all participants to kindly take note of the quiz link and e-certificate link. After giving the quiz, the certificate will be available to download. So the quiz forum will be open for half an hour. So please, it is requested to all participants to give the quiz and the feedback. And after that, you can download the certificates after 2 hours. So please take the note of the links, or you may take the screenshot. We are closing the session within 1-2 minutes.
Thank you, Mr. Prem. Thank you for your support.
Thank you, sir. Thank you so much. Thank you.
Thank you, Som. Thank you.
Thank you
Bye.
so much, sir.
Thank you, sir.
Bye, sir.
Thank you so much.
Sir, we may now close the session, sir.
Yeah.
The participants'
Obviously.
number has been decreased. I think they have taken the links.
So I'm stop sharing the screen.
Yes. Already the participant can take the screenshot.
Thank you, sir.
Thank you so much.
Thank you all participants.